There is a list of 5. 5 times in the history of this planet, life walked to the edge of the abyss and looked down. Everyone knows the third one on that list, the one that ended the dinosaurs, the one with the asteroid, the one that left a crater 180 kilometers wide in what is now Mexico. Many people know the second one, the great dying at the end of the Permian, the one that killed 96% of all species and came closer to sterilizing this planet than any other event in the history of complex life. But the first one on that list, the one that started everything, the one that established the template for how a world ends, almost nobody knows. It happened 443 million years ago. It killed 85% of all species on Earth. And unlike most of the others, it did not have one cause. It had two. 2 separate blows, separated by millions of years, arriving from different directions, hitting different vulnerabilities. The first one froze the planet. The second one drowned what was left. This is the story of the first end of the world. But before we get to the ending, we need to understand what was being ended. And to do that, we need to go back further, to the world that existed before the catastrophe, to a planet so different from the one we inhabit that describing it requires a deliberate effort to set aside almost every assumption we carry about what Earth looks like and how it works. Before we go further, if you find this kind of deep history compelling, please consider subscribing to this channel. It genuinely helps us continue making work like this. The Ordovician period began approximately 485 million years ago, at the conclusion of the Cambrian, and it lasted until 443 million years ago, a span of roughly 42 million years. 42 million years is a number that resists intuition. For comparison, the entire history of primates, from the first small tree-climbing mammals that gave rise to our lineage to the present moment, spans approximately 65 million years. The Ordovician alone was 2/3 as long as the entire primate evolutionary story, and it was packed in its marine environments with biological innovation of a pace and scale that would not be matched again for a very long time. To understand the Ordovician, you first have to understand what it inherited from the Cambrian, and what the Cambrian had bequeathed to it was both spectacular and, by the standards of what was coming, relatively simple. The Cambrian explosion, that remarkable burst of animal diversification that occurred roughly 540 million years ago, had produced the basic architectural blueprints of most major animal groups. The fundamental body plans, the general solutions to the engineering problems of being an animal, had been established. But the Cambrian world, for all its revolutionary novelty, was, in ecological terms, relatively thin. The food webs were short, the niches were few, and the total diversity of species, while extraordinary compared to anything that had existed before, was modest compared to what was about to develop. The transition from the Cambrian to the Ordovician was not marked by a dramatic extinction event of the kind that would characterize later period boundaries. It was more of a gradual transition, with some Cambrian groups declining and others expanding, and with the overall trajectory of life pointing clearly upward in terms of diversity and ecological complexity. What happened next is called the Great Ordovician Biodiversification Event, and it is one of the most important and least discussed episodes in the history of life on this planet. Over the course of the Ordovician period, the number of marine animal genera increased by a factor of approximately 3. This is not a small number. Tripling the diversity of life in 40 million years represents one of the fastest sustained expansions of biological complexity in the geological record. This diversification was not random. It followed specific patterns, filled specific ecological spaces, and produced specific innovations that would shape the trajectory of life for hundreds of millions of years afterward. Understanding why it happened requires understanding the physical world in which it took place. The geography of the Ordovician Earth was profoundly different from anything familiar to us. The planet had no polar ice caps at the beginning of the period, or rather, it had no permanent ice. The climate was warm, warmer than today across most latitudes, and the oceans were high, covering vast areas of the continental interiors in shallow, warm, sunlit seas. The arrangement of the continents bore no resemblance to anything on a modern map. The dominant geographic feature of the Ordovician world was a supercontinent called Gondwana, an enormous landmass that incorporated what would eventually become South America, Africa, Antarctica, Australia, India, and the Arabian Peninsula, assembled into a single continental block of staggering size. Gondwana was not located where any of these modern continents are now. In the early Ordovician, Gondwana was positioned with its center roughly over the equator. But as the period progressed, it drifted southward in a movement that would eventually have catastrophic consequences. Surrounding Gondwana were smaller land masses, fragments of what had been earlier supercontinents, now scattered across the global ocean. Laurentia, the ancient core of what would eventually become North America, was positioned near the equator, rotated roughly 90 degrees from its modern orientation, its broad interior flooded by shallow tropical seas. Baltica, the ancient nucleus of northern Europe, was drifting northward from its early Ordovician position in southern temperate latitudes. Avalonia, a small terrain that would eventually contribute to the coastlines of eastern North America and western Europe, was separating from Gondwana and beginning its drift northward. Between these land masses, ancient oceans spread. The Iapetus Ocean, the predecessor of the Atlantic, separated Laurentia from Baltica and Gondwana. The Rheic Ocean was beginning to open as Avalonia moved away from Gondwana. The geography was dynamic, in constant, slow motion, driven by the same plate tectonic processes that operate today, but producing a planetary arrangement that would have been unrecognizable to any modern observer. The climate of this world was, for most of the Ordovician period, significantly warmer than today. Carbon dioxide concentrations in the atmosphere were substantially higher than modern levels, perhaps 8 to 15 times higher by some estimates, driving a greenhouse effect that kept global temperatures elevated. The evidence for this comes from multiple sources. The distribution of evaporite minerals, which form only in warm, arid conditions. The presence of tropical carbonate reef systems at relatively high paleolatitudes. The isotopic composition of fossil shells, which records the temperature of the seawater in which the organisms lived. By all of these measures, the Ordovician world was a warm world, particularly in its earlier phases, and this warmth was a key driver of the extraordinary biological productivity that characterized the period. The shallow seas that covered the continental shelves and interiors were the engine of Ordovician life. Sunlit, warm, rich in dissolved minerals washed from the surrounding land by chemical weathering, these environments were extraordinarily productive, and they became the site of the most rapid and sustained diversification of marine animal life that the planet had yet seen. The diversity of life forms filling these seas by the Middle Ordovician was breathtaking in its range, from microscopic plankton to meter-scale predators, from filter-feeding organisms that barely moved to active swimmers pursuing prey across open water. Trilobites, those iconic arthropods that had been the dominant complex animals of the Cambrian, remained important members of the Ordovician fauna, but they were no longer the only show in town. They had diversified enormously from their Cambrian ancestors, producing new body forms adapted to new ecological roles. Some Ordovician trilobites were blind, living in deep, dark water where eyes were useless. Others had enormous, sophisticated compound eyes capable of detecting predators from considerable distance. Some were active swimmers, others ploughed through soft seafloor sediments in search of organic matter. The trilobite body plan, essentially an arthropod chassis with a segmented exoskeleton, proved flexible enough to be adapted to an enormous range of ecological niches, and the Ordovician saw trilobite diversity reach its all-time peak. But trilobites were just one component of the Ordovician ecosystem. Brachiopods, a group of filter-feeding marine invertebrates that superficially resemble clams but are in fact quite unrelated to them, exploded in diversity during the Ordovician to become one of the dominant groups in many shallow water communities. Unlike clams, which are mollusks and filter water by passing it over gills, brachiopods filter water using a specialized feeding organ called a lopophore, a crown of ciliated tentacles that creates water currents and traps food particles. Brachiopods attach themselves to hard substrates, to each other, and to reef-forming organisms, building dense communities on the sea floor that in ecological terms resembled the mussel beds and oyster reefs of modern coastal environments. By the Middle Ordovician, brachiopod diversity had increased to a level that would not be substantially exceeded until the Devonian, and the sheer abundance of brachiopod shells in Ordovician rocks is one of the most striking features of the period's fossil record. Echinoderms, the group that today includes sea stars, sea urchins, sea cucumbers and crinoids, were undergoing their own remarkable diversification. The Ordovician seafloor was populated by crinoids, filter-feeding echinoderms that superficially resemble plants, with a long stalk anchored to the substrate and a crown of feathery arms that collected food particles from passing water currents. Crinoids formed dense meadows on the Ordovician seafloor, particularly in areas with moderate current flow that brought a steady supply of suspended organic matter. Their remains, the distinctive segmented plates of their stalks and the cup-shaped structures that held their feeding arms, are enormously abundant in Ordovician limestone deposits worldwide, in some cases forming rock composed almost entirely of crinoid debris. Alongside the crinoids lived a diversity of other echinoderm groups, many of them now entirely extinct, including blastoids, cystoids, and various other filter feeders with body plans that no longer exist in any form in the modern world. Bryozoans, colonial filter feeders that build intricate calcium carbonate frameworks, appeared in the fossil record at the very beginning of the Ordovician and diversified rapidly through the period. Ordovician bryozoan colonies took a remarkable variety of forms, from flat encrusting sheets covering hard surfaces to elaborate branching structures that grew upward from the sea floor, creating three-dimensional habitat complexity that other organisms could exploit. Bryozoans became important reef builders in the Ordovician, participating alongside corals and other organisms in creating the first complex reef ecosystems in Earth's history. The appearance of true corals in the Ordovician is one of the period's most significant biological innovations. Tabulate corals and rugose corals, both now extinct, were the dominant reef-building corals of the Paleozoic era, and their first appearance in the Ordovician fossil record marks the beginning of a reef-building tradition that, with modifications and with the corals themselves being replaced by entirely different groups, has continued to the present day. Ordovician reefs were not identical in their ecological structure to modern coral reefs. The dominant reef builders were different, the associated fauna was composed of entirely different species, and the physical structure of the reefs reflected the specific organisms that built them. But functionally, Ordovician reefs served many of the same ecological roles as modern reefs. They created structural complexity. They concentrated biodiversity. They provided habitat for a huge variety of organisms, and they were centers of intense biological activity and productivity. Above the sea floor and in the open water, the Ordovician oceans were populated by a remarkable diversity of swimming and floating organisms. Graptolites, colonial animals that built V-shaped or multi-branched colonies of tiny individual organisms, were one of the most abundant planktonic groups of the Ordovician. Graptolite colonies floated in the open ocean, attached to gas-filled flotation devices, and when they died, their remains sank to the sea floor and were preserved in enormous numbers in fine-grained marine sediments worldwide. Graptolites are among the most useful fossils for dating Ordovician rocks because they evolved rapidly, and different species are characteristic of specific, geologically brief time intervals. The graptolite fossil record provides the primary framework for subdividing the Ordovician into its constituent stages and zones, and understanding graptolite evolution is central to understanding Ordovician biostratigraphy. Among the most dramatic creatures of the Ordovician seas were the nautiloids, cephalopod mollusks related to the modern nautilus, but in some cases reaching sizes that dwarfed anything in the modern ocean. The largest Ordovician nautiloids, belonging to groups like Camaraoceras, had straight, conical shells that could reach lengths of 9 meters or more, making them among the largest animals on Earth during the period. These animals were active predators, using their tentacles to seize prey and their jet propulsion to move through the water. As the dominant large predators of the Ordovician seas, they occupied an ecological role somewhat analogous to that of large sharks in modern marine ecosystems, controlling prey populations and influencing the ecological structure of the communities below them in the food web. The existence of large, active predators like the giant nautiloids is itself evidence for the ecological complexity of the Ordovician marine world. Large predators can only exist in ecosystems that are productive enough to support the food chains that lead to them. A 9-meter nautiloid required an enormous amount of prey to sustain itself, which required an enormous amount of prey's prey, and so on down through the food web to the primary producers at the base. The presence of apex predators of this size is testimony to the productivity and ecological complexity of the Ordovician marine ecosystem, which, by the middle of the period, had become the most complex and diverse ecosystem that had ever existed on this planet. The vertebrates, the group to which we ourselves belong, made their first significant appearance in the marine fossil record during the Ordovician. The earliest vertebrates were jawless fish, members of a group called agnathans, which included the ancestors of the modern lampreys and hagfish, as well as a vast diversity of armored forms that have no living relatives. Ordovician vertebrates were small, typically a few centimeters to perhaps 20 or 30 centimeters in length, and they were covered in armor made of mineralized bone plates that protected their bodies from predators. They lacked jaws, and most of them were probably filter feeders or bottom dwellers that consumed organic material from the seafloor sediment. They were not the dominant animals of their ecosystems, not by a long way. But their appearance in the Ordovician fossil record marks the beginning of the vertebrate story, the beginning of the lineage that would eventually produce fish and amphibians and reptiles and mammals and, eventually, us. The absence of life on land during the Ordovician is one of the most striking features of the period, and it requires some explanation, because the land was not completely lifeless. It was simply almost completely lifeless, and the life that existed there was primitive, microscopic, or near microscopic, and confined to specific microenvironments where the conditions were survivable. The reason for the near-total absence of life on land was the absence of an ozone layer in the upper atmosphere. Today, the ozone layer, a region of the stratosphere where ozone molecules are concentrated, absorbs the great majority of the ultraviolet radiation arriving from the sun before it reaches the surface. Without this protective shield, ultraviolet radiation at the surface would be intense enough to damage or destroy the DNA of most organisms within minutes of exposure. The ozone layer is produced by the photochemical interaction of oxygen molecules with ultraviolet radiation, a process that requires atmospheric oxygen at concentrations significantly above the trace levels that had existed for most of Earth's history. During the Ordovician, atmospheric oxygen was rising, driven by photosynthesis in the marine and freshwater environments, but it had not yet reached the levels required to produce an ozone layer thick enough to provide meaningful protection to land-dwelling organisms. In this context, the evolution of terrestrial life during the Ordovician was not a march of conquest across open land, but a careful, tentative exploration of marginal environments where moisture, shade, or other local conditions provided some shelter from the killing ultraviolet flux. The most successful colonizers of the Ordovician land surface were organisms with robust protective coverings, or with the ability to dry out and revive, the same biological strategies that allow modern organisms like tardigrades and certain mosses to survive extreme conditions. Primitive plants related to modern liverworts and mosses appear to have established themselves on moist terrestrial surfaces near water bodies during the Ordovician, forming thin biological crusts that stabilized soil and created microenvironments slightly more hospitable than bare rock. Fungi, which had probably made the transition to land earlier, were also present, and some evidence suggests that the symbiotic relationship between plants and fungi that underlies most modern terrestrial plant communities was already being established during this period. The contribution of these early terrestrial organisms to the global carbon cycle was not negligible. Plant-like organisms on land engaged in photosynthesis, consuming carbon dioxide and releasing oxygen. Their roots, or rather the root-like structures called rhizoids that served a similar function in these primitive plants, accelerated the chemical weathering of rocks, releasing minerals into runoff water that eventually reached the ocean, where it fertilized marine productivity. The early terrestrial biota of the Ordovician was, in this sense, a geochemical force as well as a biological one, subtly modifying the chemistry of the atmosphere and ocean through its collective activity. These modifications would eventually contribute to the climatic changes that defined the end of the period, though the precise mechanisms and magnitudes remain subjects of active scientific debate. The first arthropods to make tentative excursions onto land during the Ordovician were probably myriapod-like creatures related to the ancestors of modern millipedes and centipedes. The evidence for this is fragmentary and indirect, consisting primarily of trace fossils, the tracks, burrows, and other physical evidence of organism activity rather than body fossils of the organisms themselves. Interpreting Ordovician terrestrial trace fossils is challenging because many of the organisms that made them had no hard parts to preserve as body fossils, and the traces themselves can be ambiguous in their interpretation. What seems clear is that the land surface, while inhospitable to most life, was not completely unvisited by animals during the Ordovician, and that the foundation for the eventual conquest of the land that would occur in the Silurian and Devonian was being laid, slowly and tentatively, during this period. The chemical environment of the Ordovician Ocean was significantly different from that of the modern ocean in ways that affected both the kinds of organisms that could thrive and the kinds of skeletons and shells they could build.
The ratio of magnesium to calcium in seawater influences whether organisms preferentially precipitate calcite or aragonite, the two common crystal forms of calcium carbonate. Ordovician seawater had a relatively high calcium concentration and relatively low magnesium concentration, creating what geochemists call a calcite sea, in which organisms building carbonate skeletons tended to produce calcite rather than aragonite structures. This influenced the kinds of reef-building organisms that were ecologically dominant because different organisms preferred different carbonate minerals. And it affected the preservation of fossils because calcite is more stable in the geological record than aragonite. The temperature structure of the Ordovician Ocean also differed significantly from the modern ocean. Today, the deep ocean is cold, fed by dense, cold water that sinks in the polar regions and flows along the ocean floor toward the equator. This thermohaline circulation, driven by temperature and salinity differences, creates a globally connected pattern of ocean currents that distributes heat, nutrients, and oxygen throughout the world ocean. In the warm Ordovician world of the early and middle parts of the period, with no polar ice caps to generate cold, dense water, the deep ocean was likely warmer and more poorly oxygenated than today, because the density contrast between surface and deep water was smaller, driving weaker circulation. The distribution of oxygen in the ocean, controlled by this circulation, influenced which parts of the seafloor could support active communities of bottom-dwelling organisms, and which were effectively dead zones where only anaerobic bacteria could survive. Understanding the Ordovician ocean circulation is important for understanding both the success of Ordovician life and the nature of the catastrophe that eventually ended it. Because the transition from a warm, stratified ocean to a cold, glaciated world involved a dramatic reorganization of ocean circulation, with consequences that reached every corner of the marine ecosystem.
By the Middle Ordovician, approximately 460 million years ago, the marine ecosystem had reached a level of complexity and diversity that the planet had never before seen. The Great Ordovician biodiversification event was in full swing, and the oceans teemed with life at every level of the food web, from microscopic phytoplankton and zooplankton at the base to giant nautiloid predators at the apex, with dozens of ecological levels and thousands of species in between. The shallow tropical seas around Laurentia, Baltica, and the northern margin of Gondwana were particularly rich, their warm, clear, sunlit waters supporting dense communities of corals, bryozoans, brachiopods, crinoids, trilobites, and 100 other groups. These communities were not static. They changed over time as species evolved and went locally extinct, as sea level rose and fell in response to tectonic and climatic forces, as the positions of the continents slowly shifted. But the overall trajectory of life in the Middle Ordovician was upward, toward greater diversity, greater ecological complexity, and greater sophistication of the biological systems that connected organisms to each other and to their physical environment. This trajectory would continue for another 20 million years before the warning signs of what was coming began to appear.
And when they appeared, they came from the most ancient source of geological disruption available: the movement of continents and the changing chemistry of the atmosphere. Forces that operated on timescales so vast that no individual organism, no ecosystem, no lineage could perceive them or respond to them in any direct way. The world was about to change, and the change would be so total, so rapid in geological terms, and so comprehensive in its effects that almost nothing would survive it intact. The first hint of what was coming was subtle, visible only in retrospect through the careful analysis of chemical signals preserved in ancient rocks. Approximately 460 million years ago, in the Middle Ordovician, the isotopic composition of carbon in marine sediments began to shift in ways that suggested changes in the global carbon cycle. Carbon exists in two stable isotopic forms, carbon-12 and carbon-13, and the ratio between them in marine carbonates reflects the balance between the burial of organic carbon and the delivery of carbon from geological sources like volcanic activity and chemical weathering. When the ratio shifts in specific ways, it indicates that the carbon cycle is being perturbed, that something is changing in the balance of sources and sinks that normally keeps atmospheric carbon dioxide concentrations relatively stable over geological timescales. The Middle Ordovician carbon isotope excursion, as it is known in the scientific literature, has been interpreted in multiple ways, and the debate about its causes and implications remains active. One prominent interpretation connects it to a major episode of volcanic activity associated with the emplacement of what are called large igneous provinces, vast outpourings of lava from mantle hotspots that can release enormous quantities of carbon dioxide and sulfur dioxide into the atmosphere over millions of years.
The Ordovician was a period of significant tectonic activity, with the assembly and dispersal of various continental fragments producing frequent episodes of mountain building, volcanic activity, and ocean floor spreading. The volcanic gases released during these episodes would have affected the chemistry of the atmosphere and ocean in ways that could trigger biological responses, both positive by fertilizing the ocean with minerals, and negative by acidifying seawater or altering global temperatures. Another interpretation focuses on the role of the early land plants in drawing down atmospheric carbon dioxide through chemical weathering. As primitive plants spread across the land surface, their root-like structures accelerated the breakdown of silicate rocks, a process that consumes carbon dioxide from the atmosphere and releases calcium and magnesium ions into rivers that eventually carry them to the ocean. The ocean then uses these ions to build carbonate shells, effectively locking the carbon away in sediment. If the early expansion of terrestrial vegetation was sufficient to significantly accelerate this process, it could have driven a substantial drawdown of atmospheric carbon dioxide, cooling the climate and setting the stage for the glaciation that would eventually define the end of the period. This hypothesis, sometimes called the Ordovician plant hypothesis, remains controversial because the evidence for the extent of early land plant cover during the Ordovician is limited and ambiguous. But it has the advantage of connecting two major events of the period: the expansion of terrestrial life and the subsequent climate deterioration, through a plausible geochemical mechanism. There is also a more exotic hypothesis for the Ordovician extinction that deserves mention, not because it has achieved widespread scientific acceptance, but because it illustrates the breadth of mechanisms that planetary catastrophes can involve. In 2004, physicists Adrian Mellott and Brian Thomas proposed that a gamma-ray burst, an extraordinarily energetic explosion associated with the collapse of a massive star or the merger of two compact stellar remnants, occurring within a few thousand light-years of Earth, could have caused or contributed to the Ordovician extinction. A gamma-ray burst of sufficient intensity striking Earth would have triggered a cascade of atmospheric chemical reactions that would have destroyed much of the ozone layer, exposing the surface to intense ultraviolet radiation for months to years. The evidence cited in support of this hypothesis includes the pattern of extinction, which showed some preferential effect on shallow water marine organisms most exposed to ultraviolet radiation, and the direction from which the extinction effects appeared to come, which showed some evidence of asymmetry consistent with a directional burst of radiation. The hypothesis remains speculative and contested, but it has the virtue of highlighting that the Ordovician extinction was not simply a story of conventional geological forces, and that the full range of mechanisms capable of perturbing the Earth system extends beyond the familiar catalogue of volcanoes and sea level changes. Whatever the specific combination of causes that set the stage for what was coming, the physical trajectory of the Ordovician Earth in the late part of the period was moving steadily toward crisis. Gondwana was drifting southward, driven by the slow churning of the mantle beneath, and as its southern margin approached the polar regions, the conditions for continental glaciation were becoming increasingly favorable. Ice sheets form preferentially over land rather than ocean, because land surfaces cool more rapidly and retain snow more effectively than ocean surfaces. A continent positioned over or near the South Pole provides the geographical platform on which ice can accumulate. And once ice begins to accumulate, powerful feedback mechanisms amplify the initial cooling. Ice reflects sunlight back into space rather than absorbing it, reducing the amount of solar energy that reaches the planetary surface and causing further cooling, which encourages more ice formation, which reflects more sunlight, in a runaway feedback that can transform a marginally cool climate into a full glaciation with remarkable geological speed. The late Ordovician glaciation, known as the Hernantian glaciation after the Hernantian age that represents the final stage of the Ordovician period, was one of the most severe glacial episodes in the history of complex life. Estimates of the extent of the ice sheet that developed over Gondwana suggest that it was comparable in scale to the ice sheets of the most recent ice age, covering millions of square kilometers of what is now Africa and South America with ice kilometers thick. Glacial deposits from this event, distinctive sedimentary rocks called tillites that form from the debris carried and deposited by glaciers, have been found in the Sahara Desert, in the Atlas Mountains of North Africa, in the Amazon Basin of South America, and in other locations that today lie in tropical and subtropical latitudes. Evidence that these regions, now among the warmest on Earth, were once buried under thick ice. The first blow of the Ordovician extinction was delivered by this glaciation, and it struck through two related mechanisms operating simultaneously. The first was a dramatic drop in sea level. When water is locked up in continental ice sheets, it cannot flow back to the ocean, and global sea level falls in proportion to the volume of ice accumulated on land.
During the Hernantian glaciation, sea level fell by an estimated 50 to 100 meters, a change that seems modest by the standards of human construction, but is catastrophic by the standards of marine ecosystems. The shallow epicontinental seas that had been the most productive and diverse environments of the Ordovician world, the warm, sunlit, biologically rich seas that had covered vast areas of Laurentia, Baltica, and the tropical margins of Gondwana, simply ceased to exist as the sea level dropped below the rim of the continental shelves. Communities that had existed for tens of millions of years, building their complex ecological structures over geological time, found their physical environment disappearing beneath them. The seafloor that had been habitat became dry land. The organisms that had lived there had nowhere to go. The second mechanism of the first blow was the temperature drop itself. The warm water communities of the Ordovician, adapted over millions of years to the specific temperature regime of the Middle Ordovician Ocean, were suddenly confronted with water temperatures significantly lower than anything their evolutionary history had prepared them for. Cold water holds more dissolved oxygen than warm water, which might seem beneficial, but cold water also changes the chemistry of carbonate minerals, making it more corrosive to the calcium carbonate shells that the vast majority of Ordovician marine organisms used to build their skeletons. Cold water changes the biology of the food web from base to top, reducing the growth rates of primary producers, altering the development times of larvae, changing the behavior and physiology of active organisms. The community of organisms that had thrived in the warm Ordovician seas was not adapted to the cold Hernantian world, and the mismatch between organism and environment was lethal on a massive scale. The extinction that resulted from the first blow was severe. Many of the characteristic groups of the Ordovician, the graptolites, the trilobites, the brachiopods, the nautiloids, the conodonts, lost enormous fractions of their diversity.
Some groups were reduced to a handful of surviving lineages. Others disappeared entirely. The ecological communities that had characterized the Middle Ordovician, those complex, densely packed assemblages of diverse organisms filling every available niche in the shallow tropical seas, were devastated. What replaced them in the cold, shallow waters of the Hernantian world was a relatively impoverished fauna dominated by a few cold-tolerant groups, particularly a distinctive assemblage of brachiopods called the Hernantia fauna, which spread across the globe as the cold conditions spread, filling the ecological vacuum left by the extinction of the warmer water communities. If the extinction had stopped there, it would already rank as one of the major crises in the history of life. But the extinction did not stop there. This is what makes the Ordovician Silurian extinction event uniquely terrible and uniquely instructive. It was not one catastrophe, but two, and the second was in some ways more damaging than the first. The Hernantian glaciation, for all its severity, was geologically brief. The astronomical and geological forces that had created the conditions for glaciation changed, and the ice sheets began to melt. Gondwana continued its complex tectonic drift, perhaps moving the continental mass slightly away from the polar position that had made glaciation possible. Volcanic activity may have released additional carbon dioxide, warming the climate. Whatever the precise trigger, the ice sheets retreated, sea level rose again, and the world began to warm. Under ordinary circumstances, the return of warm, shallow seas would have provided the ecological opportunity for the survivors of the first extinction pulse to diversify and recover, filling the vacant niches with new species descended from the survivors. But something went wrong with the recovery. The melting of the ice sheets and the return of warm surface waters did not restore the ocean to anything resembling its pre-extinction state. Instead, it triggered the second blow. And this blow came from below, from the deep ocean. And it was arguably more insidious than the freezing that had come before. When the ice sheets were present, the cold climate had driven strong thermohaline circulation, similar to the modern pattern in which cold, dense water sinks at high latitudes and flows along the ocean floor, carrying oxygen to the deep ocean. This circulation had kept the deep ocean oxygenated, or at least better oxygenated than it would otherwise have been. When the ice melted and the climate warmed rapidly, the temperature contrast between high and low latitudes decreased, reducing the density contrast between surface and deep water. And thermohaline circulation weakened or collapsed. Without the steady supply of oxygenated surface water being carried to depth by sinking, the deep ocean became anoxic, meaning it was depleted of dissolved oxygen to the point where most aerobic life could not survive there. This transition to ocean anoxia had consequences that extended far beyond the deep ocean. As organic matter sank from the productive surface waters and accumulated on the anoxic sea floor, it was decomposed by anaerobic bacteria rather than aerobic organisms, a process that releases different chemical compounds and can produce hydrogen sulfide, a highly toxic gas. The hydrogen sulfide can diffuse upward from anoxic bottom waters, creating a lethal chemical barrier that kills everything caught in its path. Upwelling currents, which normally bring nutrient-rich deep water to the surface and fertilize surface productivity, instead brought poisoned water, carrying hydrogen sulfide and depleted of oxygen. The same currents that, in a healthy ocean, deliver the nutrients that feed the food web were now delivering poison. The organisms that had survived the first extinction pulse now faced conditions that were, in some respects, worse than what they had already endured. The survivors of the glaciation, already reduced in number and ecological diversity, were confronted with an ocean in which large volumes of water were toxic, in which the chemical environment was dramatically different from anything their evolutionary history had prepared them for, and in which the ecological communities that had supported them had not yet had time to recover. The second pulse of extinction, driven by ocean anoxia and chemical poisoning, took the diversity of Ordovician life down to its lowest point. By the time the Ordovician-Silurian boundary had been crossed, approximately 85% of all marine species had gone extinct. In terms of the loss of genera, the taxonomic level above species, the loss was roughly 60%. These numbers, staggering as they are, represent an averaging across the entire marine biosphere. For some groups, the losses were much more severe. The graptolites, which had been one of the most abundant and widespread planktonic groups of the Ordovician, were reduced to a single surviving lineage. The trilobites, which had reached their peak diversity in the Ordovician, lost the vast majority of their families and never regained their former diversity. The conodonts, enigmatic tooth-bearing animals that are now understood to be early vertebrates, lost most of their diversity. The nautiloids were dramatically reduced. The brachiopods, while surviving in greater numbers than some other groups, lost an enormous fraction of their Ordovician diversity.
The pattern of survival was not random. It reflected the specific nature of the two extinction pulses and the specific vulnerabilities and resistances of different groups and different ecological types. Organisms living in the shallow, warm tropical seas were most severely affected by the first pulse because their preferred habitat was the one most directly destroyed by the combination of cooling and sea level fall. Organisms living in deeper water or at higher latitudes, already adapted to cooler conditions, fared somewhat better during the first pulse. But the second pulse, the anoxic event, was most severe in the mid-water and deep-water environments that had initially provided some refuge, inverting the pattern of vulnerability and ensuring that no single refuge was safe from both phases of the extinction. The survivors were a disparate and, in some ways, unpredictable group. The nautiloids were reduced but not eliminated and would diversify again in the Silurian. The cephalopods, more broadly, retained enough diversity to eventually give rise to the ammonites, one of the most successful and diverse groups of the entire Paleozoic era. The corals were severely hit but survived, their Silurian descendants rebuilding the reef communities that would become characteristic of the Middle Paleozoic. The brachiopods retained enough diversity to support a recovery that would eventually make them the dominant benthos of the Silurian and Devonian. The vertebrates, still a minor component of the Ordovician marine ecosystem, appear to have passed through the extinction relatively unscathed, or at least to have lost fewer lineages than many more prominent groups. This survival would prove consequential, though no Ordovician organism could have known it. The lineages that survived
would eventually give rise to the jawed fissures of the Silurian and Devonian. And from jawed fissures would come every terrestrial vertebrate that has ever lived, including the species reading these words. The recovery from the Ordovician Silurian extinction was, by geological standards, relatively rapid. Within 5 to 10 million years of the extinction, Silurian marine communities had recovered to levels of diversity comparable to those of the early Ordovician, and the subsequent Silurian and Devonian periods would see continued expansion of marine diversity. But the recovery was not simply a restoration of the Ordovician world. The specific groups that had dominated the Ordovician marine ecosystem did not simply regrow from their surviving remnants into their former configurations. Instead, the vacant ecological space left by the extinction was filled by new groups, new combinations of organisms, new ecological structures that differed in important ways from what had come before. The Silurian world was not the Ordovician world restored. It was something new, built on the ruins of what had come before, shaped by the specific pattern of what had survived and what had not. This pattern of extinction followed by recovery, followed by a new and different world rather than restoration of the old one, is one of the most important generalizations to emerge from the study of mass extinctions. Extinctions do not simply reset the clock, returning life to an earlier state from which it then reruns the same trajectory. They redirect the trajectory, opening new possibilities by eliminating old occupants of ecological space and closing other possibilities by eliminating lineages that might have given rise to different evolutionary futures. The Ordovician extinction redirected the trajectory of life in specific ways that shaped the entire subsequent history of animal evolution. Though tracing the precise causal chains from a late Ordovician extinction pulse to any specific feature of modern life requires more paleontological detective work than has yet been done. What the Ordovician extinction teaches us about the modern world is a question that paleontologists and earth scientists have increasingly turned to in recent decades, as the parallels between Ordovician conditions and the trajectory of the modern Earth system have become uncomfortably apparent. The Ordovician world was characterized by high atmospheric carbon dioxide concentrations, a warm global climate, and high sea levels, conditions that shifted dramatically and catastrophically as the climate system crossed the threshold into glaciation. The modern world, driven by human industrial activity, is moving in the opposite direction along the carbon dioxide and temperature axes, rapidly warming a world that was already warm by recent geological standards. But the Ordovician example demonstrates that the relationship between carbon dioxide, temperature, and biological catastrophe is not linear and not simple. The Ordovician extinction was caused by cooling, not warming, triggered by the drawdown of carbon dioxide rather than its accumulation. What the Ordovician example most powerfully illustrates is the danger of rapid climate change in either direction, and the particular danger of transitions between climate states. It was not simply the cold that killed so much Ordovician life. It was the speed of the transition, the mismatch between the rate of environmental change and the capacity of biological communities to adapt. And it was the double nature of the catastrophe, the way in which the return to warmer conditions, which might have been expected to provide relief, instead delivered a second blow through the mechanism of ocean anoxia. The lesson is
that complex Earth systems can respond to perturbation in unexpected ways, that what appears to be a recovery can conceal the conditions for a second catastrophe, and that the biological consequences of rapid environmental change are not always predictable from the direction of that change alone. The specific chemistry of what happened to the Ordovician Ocean during and after the glaciation deserves closer examination, because it reveals mechanisms of planetary scale change that operate invisibly, beneath the surface of events that seem straightforward when viewed only from above. The ocean is not a passive container of saltwater. It is an active chemical system, continuously exchanging gases with the atmosphere, cycling nutrients through biological and geological processes, and maintaining a complex balance of dissolved compounds that makes it habitable for the organisms living within it. When that balance is disrupted, the consequences propagate through the system in ways that are difficult to predict and impossible to stop once they have gained momentum. The Ordovician Ocean, during the peak of its biological productivity in the middle part of the period, was running what geochemists call an efficient biological pump. Phytoplankton and other photosynthetic organisms in the sunlit surface waters were consuming carbon dioxide and nutrients, building their bodies from these raw materials, and when they died, their organic matter sank toward the seafloor. Some of this sinking organic matter was consumed by organisms in the water column before it reached the bottom, recycling the nutrients back into the system. But a significant fraction reached the seafloor and was buried in sediment, removing carbon from the active cycle and sequestering it in rock for geological timescales. This biological pump was one of the primary mechanisms by which the Ordovician Ocean influenced the global carbon cycle and, through it, the concentration of carbon dioxide in the atmosphere. The efficiency of this pump depended critically on the physical structure of the ocean. In a well-stratified ocean, where warm, less dense surface water sits atop cold, denser deep water with limited mixing between the two layers, the biological pump operates most effectively, because nutrients that sink out of the surface layer are not quickly returned, and organic carbon exported to depth has a higher probability of being buried rather than recycled. The warm Ordovician Ocean of the early and middle periods was likely highly stratified, running an efficient biological pump, drawing down atmospheric carbon dioxide and gradually cooling the climate over millions of years. This is one mechanism by which the conditions for the eventual glaciation may have been created, slowly and invisibly, by the biological activity of the organisms that would eventually be destroyed by its consequences. When the glaciation began and the climate shifted rapidly toward cold, the physical structure of the ocean changed dramatically. Cold polar waters, denser than the warm tropical waters they were displacing, began to drive stronger thermohaline circulation. The previously stratified ocean began to overturn, bringing deep water to the surface and surface water to depth. This overturn was, initially, not entirely negative for marine life. The upwelling of deep water brought nutrients to the surface, potentially stimulating biological productivity in some regions, even as the cooling temperatures and falling sea levels were devastating shallow water communities elsewhere. The pattern of winners and losers during the first pulse of the extinction was partly determined by this complex interplay between cooling, sea level change, and changing ocean circulation. But the same circulation that brought some nutrients to the surface during the glaciation was also setting up the conditions for the second
and in many ways, more insidious phase of the catastrophe. As the ice sheets grew and the climate became increasingly cold, the chemistry of the deep ocean was changing. Cold, well-oxygenated water flowing from polar regions was ventilating the deep ocean, keeping it aerobic, supporting the organisms that lived on and just above the sea floor in deep water. This deep ocean oxygenation was maintaining the conditions that allowed organic matter reaching the sea floor to be processed by aerobic decomposers rather than accumulating in anaerobic sediments. When the ice melted and the climate warmed rapidly, this pattern reversed with startling speed. The influx of meltwater from the retreating ice sheets added a layer of fresh, cold, light water to the ocean's surface at high latitudes, reducing the density contrast that had been driving thermohaline circulation. As circulation weakened, the supply of oxygenated surface water to the deep ocean declined. Organic matter continued to rain down from the productive surface, consuming oxygen as it decomposed, but the oxygen was no longer being replenished by circulation. The deep ocean began to go anoxic, first in the most isolated basins where circulation had always been weakest, then spreading progressively into shallower and more connected regions as the anoxia intensified. The geochemical signature of this anoxic event is preserved in the chemical composition of Ordovician Silurian boundary rocks worldwide.
Isotopic ratios of sulfur in ancient pyrite. Ratios of certain trace metals, like molybdenum and vanadium, that are preferentially removed from seawater under anoxic conditions. And the carbon isotopic composition of organic matter in the sediments all tell the same story. The ocean became severely oxygen depleted over large volumes at and around the Ordovician Silurian boundary. And this oxygen depletion was both widespread and sustained. Some sections show evidence for the presence of hydrogen sulfide in the water column, the signature of eosinic conditions in which sulfate-reducing bacteria generate toxic hydrogen sulfide in anoxic bottom waters. These eosinic conditions are among the most lethal environments in the ocean, capable of killing most aerobic marine life within hours of exposure. The combination of falling sea level, cooling temperatures, and ocean anoxia that characterized the Hernantian stage created what paleontologists sometimes describe as a perfect storm of extinction drivers, each one alone capable of causing significant biological damage, but all three operating simultaneously and interactively in ways that amplified their individual effects. A species reduced by habitat loss from sea level fall was less able to withstand the additional stress of temperature change. A community already disrupted by temperature stress was less resilient in the face of chemical poisoning from expanding anoxia. The redundancy and resilience that characterize healthy, diverse ecological communities, the ability to lose some species while retaining overall function, was eroded progressively by each successive stressor, until communities that might have survived any one of the pressures in isolation could not survive their combined assault. The geographic pattern of the extinction was not uniform, and understanding the variation in extinction intensity across different regions and different environments has been one of the major research programs in Ordovician paleontology over the past 3 decades. Tropical, shallow water communities were among the hardest hit by the first extinction pulse, as might be expected given that they were most directly dependent on the warm, shallow seas that disappeared as climate cooled and sea level fell. High-latitude communities,
already adapted to cooler conditions, showed more resilience during the first pulse, but were not spared the second. Deep water communities, which might have been expected to be somewhat buffered from the surface effects of cooling and sea level change, were devastated by the anoxic event. The net result was that no marine environment, from the shallowest tropical reef to the deep open ocean, was spared significant biological damage. The pace of the extinction is a question of considerable scientific importance and ongoing debate. In geological terms, the Ordovician Silurian extinction was rapid, concentrated within an interval of perhaps 1 to 2 million years at the end of the Hernantian stage. But 1 to 2 million years is not instantaneous, and within that interval, the two pulses of extinction were separated by an uncertain but non-trivial period of time, perhaps several 100,000 years, during which the climate was in transition between the glacial maximum and the post-glacial warming. For the organisms living through this transition, the pace would have varied enormously depending on where they lived and which extinction mechanism was affecting them most directly. The fall in sea level, for example, would have occurred over thousands to tens of thousands of years, fast enough to eliminate habitats within the lifespan of many marine ecosystems, but slow enough that organisms with sufficient dispersalability and ecological flexibility might track shifting suitable conditions across the landscape or seascape. The anoxic event, by contrast, may have expanded much more rapidly, potentially poisoning large volumes of ocean within centuries to millennia, a pace that would have left little time for biological adaptation. One of the most revealing aspects of the Ordovician extinction is what it tells us about the relationship between biological diversity and ecological resilience. The Middle Ordovician Ocean, with its extraordinary diversity of species and its complex ecological structure, was in many ways a more robust and resilient system than the simpler communities of the early Cambrian. High diversity provides redundancy, multiple species filling similar ecological roles so that the loss of any single species does not collapse the function it performed. Complex ecological networks distribute energy and materials through many pathways, providing alternative routes when any single pathway is blocked. In normal times, this complexity and diversity provided the Ordovician ecosystem with considerable resistance to disruption. But the Ordovician extinction reveals the limits of biological resilience in the face of sufficiently large and rapid physical changes. When the physical environment changes faster than biological communities can adapt, when multiple stressors arrive simultaneously and interact to amplify each other's effects, and when the changes are global rather than local, eliminating the refuges from which recovery might otherwise proceed, even the most diverse and ecologically complex communities can collapse. The lesson is not that diversity is unimportant for resilience. It clearly is important. But that there are thresholds beyond which no amount of biological complexity can buffer a system against physical changes of sufficient magnitude and speed. The recovery from the Ordovician extinction in the early Silurian period was characterized by a fascinating biological phenomenon that paleontologists call the Lazarus effect. Named after the biblical figure raised from the dead, Lazarus taxa are groups that disappear from the fossil record during an extinction event and then reappear in post-extinction sediments, apparently having survived somewhere during the interval when they were absent from the record. During the Ordovician Silurian extinction, many groups seem to vanish from the fossil record at the extinction horizon, and then reappear in early Silurian rocks. This pattern reflects not true extinction and re-evolution, which would be biologically impossible, but rather survival of small, geographically restricted populations in refuges that were not sampled by the fossil record, followed by recolonization of broader areas as conditions improved. The existence of Lazarus taxa during the Ordovician recovery tells us something important about the nature of extinction and survival at the species level. Even during catastrophic global events, the geographic heterogeneity of the Earth's surface means that some locations experience conditions less extreme than the global average, providing refuges where species can persist in small numbers even as they disappear from broader areas. The size and accessibility of these refuges and the ability of survivors to disperse from them during recovery are critical determinants of how quickly and completely a biosphere can recover from mass extinction.
The Ordovician recovery, which reached pre-extinction diversity levels within approximately 5 to 10 million years, was relatively rapid by the standards of mass extinctions, suggesting that the refuges were sufficient to preserve a diverse enough set of surviving lineages to rebuild complex communities without requiring the evolution of major new body plans. The early Silurian recovery also saw some surprising ecological innovations. Groups that had been minor components of Ordovician communities suddenly becoming dominant in post-extinction environments. The nautiloids, reduced to a few lineages by the extinction, diversified again and remained important predators in Silurian seas. Jawed fish, which had been present but ecologically minor in the late Ordovician, began their diversification during the Silurian and would eventually transform the structure of marine ecosystems. Vascular plants, building on the foundation laid by the primitive bryophyte-like plants of the Ordovician, began their conquest of the land during the Silurian, fundamentally changing the terrestrial environment and setting the stage for the much more extensive colonization of land that would occur in the Devonian. The extinction had cleared the ecological stage, and the new caste assembling on it would eventually produce the world of the Devonian, sometimes called the Age of Fishes, and then the world of the first forests, and then the first amphibians stepping onto land, and then, much later, everything that followed. The study of the Ordovician period and its ending extinction has been transformed in the past 2 decades by improvements in geochemical analytical techniques that allow researchers to extract detailed environmental information from ancient rocks with a precision that was previously unattainable. Isotope ratio measurements of unprecedented accuracy, trace element analyses revealing subtle changes in ocean chemistry, organic molecular fossils preserved in ancient sediments, and high-resolution dating techniques that can pin down the timing of events to within tens of thousands of years, have collectively produced a much more detailed and nuanced picture of Ordovician environmental change than existed even a generation ago. This new data has resolved some long-standing debates while opening new ones, and has made it clear that the Ordovician world and its extinction were even more complex than earlier, coarser analyses had suggested. Particularly significant has been the improvement in understanding the relationship between volcanic activity and the Ordovician climate system. A massive episode of volcanic activity associated with the breakup of continental margins in the early and middle Ordovician, producing an interval of extraordinarily high rates of ocean floor spreading and associated outgassing of carbon dioxide,
may have been responsible for the high atmospheric carbon dioxide concentrations and warm climate of the early Ordovician. As this volcanic episode waned in the late Ordovician, the removal of its carbon dioxide source, combined with the ongoing drawdown of carbon dioxide by chemical weathering and biological productivity, would have contributed to the gradual cooling that preceded the glaciation. This volcanic hypothesis connects the tectonic history of the Ordovician to its climate history in a coherent way, and it has the advantage of explaining not just why the climate was warm in the early Ordovician, but why it cooled in the late Ordovician, without requiring a single triggering event.
The integration of these geochemical and geological insights with the biological data from the fossil record has produced what is increasingly recognized as one of the most comprehensive and mechanistically detailed accounts of any mass extinction in the geological record. The Ordovician Silurian extinction, once the most poorly understood of the Big Five, is rapidly becoming one of the best understood. And the insights it provides into the mechanisms of global biological catastrophe are of relevance far beyond the specialist community of Ordovician paleontologists. In a world increasingly concerned with the consequences of rapid environmental change, the Ordovician offers a natural experiment in planetary-scale disruption, conducted over millions of years, preserved in the rock record, and now readable with analytical tools of sufficient precision to reveal its mechanisms in detail. What that natural experiment tells us is simultaneously sobering and, in a strange way, reassuring. Sobering because the mechanisms that destroyed 85% of Ordovician species, rapid climate change, ocean chemistry disruption, habitat loss, and the complex interactions between these stressors, are not mechanisms unique to 443 million years ago. They are mechanisms that operate whenever the Earth system is pushed rapidly from one state to another. And they are mechanisms that human activity is currently engaging with unprecedented speed. Reassuring in a deeply qualified way, because the Ordovician extinction, catastrophic as it was not the end. Life survived. Not unchanged, not undiminished, not without losing things of irreplaceable value. But it survived, and in surviving it found ways to rebuild, to diversify, to fill the empty world with new forms and new complexity. The resilience of life over geological timescales, its ability to recover from catastrophes that destroy the majority of its species, is one of the most profound facts in natural history. But that reassurance comes with an essential caveat. The geological recovery from the Ordovician extinction took millions of years. For the ecosystems, the species, the lineages that did not survive, there was no recovery. The resilience of life over geological timescales is cold comfort for the timescales of human civilization, of human lives, of the specific and irreplaceable organisms whose extinction today contributes to a biodiversity crisis that some researchers are already comparing to the Big Five. The Ordovician tells us that life will go on. It does not tell us that everything currently alive will go on with it. And it does not tell us that the world on the other side of a mass extinction will be anything we would recognize or value. The first end of the world was survived, but survival and flourishing are not the same thing, and the distance between them, measured in lost species and collapsed ecosystems, and millions of years of slow reconstruction,
is one of the most important lessons that the Ordovician has to teach. The Ordovician Silurian extinction is the least studied and least publicly known of the 5 major mass extinctions, a status that reflects in part the difficulty of working with Ordovician rocks and fossils, many of which are poorly preserved or buried under younger formations, and in part the absence of the charismatic megafauna that makes other extinction events more immediately compelling to the public imagination. There are no Ordovician dinosaurs, no Ordovician mammals, nothing that looks remotely familiar to a modern viewer. The organisms that went extinct were mostly invertebrates with exotic names, creatures that most people have never heard of and cannot easily visualize.
The world they inhabited was so alien to modern experience that describing it requires sustained effort even for professional scientists. But the Ordovician extinction matters. It matters because it was the first time that complex animal life on this planet was pushed to the edge of annihilation. And because the way in which it happened established patterns and precedents that would recur in subsequent extinctions. It matters because the survivors of the Ordovician extinction were the ancestors of most of the animal groups that populate the modern world, including our own vertebrate lineage. It matters because the Ordovician world, in its brief and brilliant flourishing, demonstrated what life can achieve when conditions are favorable, building levels of biological complexity and diversity in 40 million years that took earlier life hundreds of millions of years to approach. And it matters because the mechanisms that destroyed it, rapid climate change, ocean chemistry shifts, habitat destruction on a global scale, are mechanisms that remain active today, driven now not by the slow movement of continents, but by the rapid activity of a single species that has only recently begun to understand the systems it is perturbing. 443 million years ago, the most complex and diverse marine ecosystem in the history of life was destroyed by a combination of cold and poison, by the freezing of the planet and the suffocation of its seas. The world that emerged from that catastrophe was impoverished, simplified, reduced in its diversity and its ecological complexity. But it was not dead. And in the Silurian period that followed, life did what it has always done after catastrophe. It found the open spaces. It exploited the empty niches. It built new communities from the survivors of the old. And it began, slowly and tentatively, but with the inexorable persistence that characterizes biology on geological timescales, to build a new world. That new world would eventually produce fish with jaws. And from fish with jaws would come the first vertebrates to walk on land. And from them would come everything that has ever lived on the surface of this planet, including us. The first end of the world was not the end of life. It was, in retrospect, a beginning. But that is a perspective only available to those who can look back across 443 million years and see the whole arc of the story. For the organisms that lived through it, the ones that did not make it, and the ones that barely did, there was no arc and no perspective. There was only the cold, and then the poison, and then the long silence
of the Hernantian. And then, for the survivors, the slow and uncertain business of trying again.